Welcome back to my YouTube channel. So today I'm going to discuss in detail about the molecular mechanism of apoptosis. This might come as a sub-question in your LEQ, and the previous video I have discussed the other two subheadings under the apoptosis topic. This will be the last final part. Okay. Now discuss in detail about the molecular mechanism of apoptosis. So we have discussed what is the definition of apoptosis. We have discussed what will be the morphological and biochemical changes undergoing in apoptosis. In order to understand the biochemical changes happening in apoptosis, I told this video will be helpful. So let us get into the video. So apoptosis, it is divided into two phases. Okay, first will be the initiation phase and second will be the execution phase. So just remember, in initiation phase, the caspases needed are 8, 9, 10, or the used caspases are 8, 9, 10. What about execution? It will be 3, 6, 7. In that, 3 is the most important one. Now what are caspases? Apoptosis is a caspase dependent process. Okay, so caspases are nothing but it is cysteine residues, and it's an enzyme which will cleave the DNA like after the aspartic acid will be cleaving. That's nothing, that is caspase. We don't have to go into detail over that. Now initiation, it is subdivided into two. Extrinsic pathway is there, intrinsic pathway is there. So in extrinsic pathway, the caspases which are going to get activated are 8 and 10. Then intrinsic, it will be 9. Okay, now let's get into detail. Initiation, first I am going to discuss about in detail about the extrinsic pathway, or the other name for this pathway is called the death receptor-initiated pathway. Okay, so let's study with this diagram over here. Okay, so I'll be using my pencil. As you can see, I have drawn an activated T cell, and this is an apoptotic cell. What is apoptotic cell? A cell that is gonna die. It's ready to die. This is an activated T cell, and this apoptotic cell has a receptor. The apoptosis, in the surface of the plasma membrane, there will be a receptor. It is CD95 or FAS receptor, or tumor necrosis factor, TNF factor also another name. Anyway, CD95 or FAS receptor will be present. Now this FAS receptor will be attached to the CD95 ligand. In the activated T cell, it will be projecting the CD95 ligand. They will bind together, and this will undergo trimerization. So trimerization means it's increasing. It is getting trimerized. After that, there will be, it is can you see this FADD? This is nothing but an adaptive protein. So what about this adaptive protein? After trimerization, this adaptive protein is going to bind with the pro-caspase 8 and 10. They are going to bind with the FADD, and FADD will make it activated into caspase 8 and 10. So what is this FADD? FADD is nothing but it is FAS Associated, it is a death domain. So this is, what's the function of this? It is going to bind with this pro-caspase 8 and 10. It's going to make it activated, caspase 8 and 10. So that is about initiation, extrinsic pathway. Are you clear? We have apoptotic cell, we have activated T cell. Apoptotic cell contains a receptor, CD95, FAS, or we can say TNF1, and the ligand, CD95 ligand or FAS ligand or TNF-alpha ligand, and it will get bind together, and there will be an adaptive protein, that is FADD. It will bind with the pro-caspase 8 and 10, and it will convert it to caspase, activated caspase 8 and 10. And I have mentioned something called FLIP over here. FLIP is nothing but it is an anti-apoptotic factor. So if FLIP is acting over here, this is gonna get inhibited, and apoptosis won't be happening. It is anti-apoptotic factor. Okay. Now, the extrinsic pathway of apoptosis is initiated by extracellular signals. It involves the receptor-ligand interaction as I have shown. Now let's make into words, right? what are the steps? As I told, first will be the binding of the ligand with the receptor. Here there will be FAS L or TNF-alpha plus FAS TNF receptor. They will be binding. Second step, it will be undergoing trimerization. Third step, there will be activation of FADD. What is FADD? FAS Associated Death Domain. And there will be activation of pro-caspase 8 and 10 to caspase 8. That's for the steps of extrinsic pathway or death receptor-initiated pathway. Now let's learn about the intrinsic pathway, or it is called also mitochondrial pathway. So obviously mitochondria is gonna get involved. So suppose this is one of the cell, okay, and the cell is gonna get damaged due to various reasons. Maybe the UV rays, or the decreased growth factors, or nutritional deficiency, or hormonal changes, anything. So what the cell is going to do? It will be activating the stress sensors present. The stress sensors present in the cell are BIM, BIT, BAD, NOXA, PUMA. I like to rhyme it like this: BIM, BIT, BAD, NOXA, PUMA. Or everything together is also called the BH3 only protein. These things are called the BH3 only protein. So these things, what they are going to do? It will increase the pro-apoptotic function. Yeah, so it will increase the pro-apoptotic factor and decrease the anti-apoptotic factor. Pro-apoptotic factor, sorry for the interruption. So it will increase the pro-apoptotic factor, which will give a signal to this organ. What is this? It's a mitochondria. Just forgive my drawing. And so this signal will be reaching the mitochondria, and the mitochondria will be releasing, the permeability of the mitochondria is going to increase. It will be releasing the cytochrome C. So what is the cytochrome C? It is going to get attached with the APAF1. What is APAF1? Apoptosis activating factor. APAF1, and these together is called the apoptosome. What is it called? Apoptosome. It's not visible. APOTOSOME, apoptosome. So what this is going to do? This is going to activate pro-caspase 9 to caspase 9. That's it. Before coming to the steps, I would like to tell you about this BCL2 family. What are these and what are the function of these? It can be divided into three categories. One is the anti-apoptotic, then it is a pro-apoptotic, then regulated apoptosis initiator. So what do you mean by anti-apoptotic? Obviously, it's gonna prevent apoptosis. Pro-apoptotic, it is gonna increase apoptosis, or it is gonna induce apoptosis. Regulated apoptosis, it's gonna balance it between. Okay, so the anti-apoptotic factors, you have to learn it by heart. These are the CL. You can see the CL here. BCL2, BCLXL, MCL1. Like that you have to learn. Pro-apoptotic, backs and back. Regulated apoptosis initiator, BIM, BIT, PAT, NOXA, PUMA. We already learned. Or we can also call it as the BH3 only proteins. You can also call that. Now, let's come into our topic. We will write this in a flow chart, whatever we have learned from the diagram. First I told some kind of stress. What is it? Decrease in the growth factor. Then it is going to decrease the BCL2 activation. So what do you think if the BCL2 is getting decreased? BCL2 is an anti-apoptotic factor. So anti-apoptotic factor is going to get decreased. So the prevention is decreased. That is decreased anti-apoptotic factor. Now since it is getting decreased, now these BCL2 get replaced by back and backs. What are these? These are pro-apoptotic factors. Pro-apoptotic factors, it will induce or it will increase or it will promote apoptosis, right? So this will lead to increased mitochondrial permeability, and due to this there will be increased efflux of cytochrome C from the mitochondria into the cytoplasm, and it will combine with APAF1 and it will convert the pro-caspase 9 to caspase 9. Okay. Now let's learn about our second phase, that is the execution phase. What is happening in the execution phase? Now what and all we have the activated 8 and 10. From where we got it activated from? Extrinsic pathway. 9 just now intrinsic pathway, it got activated. Now we all have all the three activated caspases, which in turn activates the 3, 6, 7, which are the execution caspases, right? And these 367 in turn help in the activation of phospholipase, protease and endonuclease, PPE. You remember, right? Phospholipase for the it will lead to membrane damage, protease cytoskeleton and endonuclease DNA fragmentation. So it's leading to cell death. Now, so in brief, if I am talking about apoptosis, I'll be saying like this. Now we have the cells and it undergone some stress and it's gonna die. So it's basically apoptotic bodies or apoptotic cells. It will be composed of cytoplasm and tightly bound organelles. It will have tightly bound organelles and maybe it have nuclear fragments or it may not have nuclear fragments. That is an apoptotic body. And these apoptotic bodies will give signal, eat me, eat me, eat me, I am not needed, just kill me, eat me signal, and it will undergo phagocytosis leading to cell death. Now these are few extra things. This cell you are seeing over here, I am naming it, this is an apoptotic body. This is an apoptotic bodies. So these apoptotic bodies will be presenting with few proteins. One will be the C1q, other thing is the thrombospondin, third one will be the phosphatidylserine. So what we can see over here, these things will give the signal, eat me, eat me, eat me, and these signals are heard by macrophages. So macrophages, what they will do, they are going to engulf this body, they are going to eat it, they are going to make it die, there will be cell death. And one more thing you have to see over here, I have written the thing called flipping of phosphatidylserine. Flipping. So what do you mean by flipping? In a normal healthy cell, the phosphatidylserine will be present over here only. So since apoptosis is happening, it will be flipping to the outer membrane. So don't you think it will be a good marker? There is a marker for flipping of phosphatidylserine, that is the annexin 5. It is for the identification of flipping of phosphatidylserine. So when we see the annexin 5, we can know that the flipping has happened. Flipping has happened means some apoptosis is going on. And I forgot to mention, this process is called efferocytosis. I will write it down. Efferocytosis. It's called efferocytosis. Now, few of the molecular markers, this CD95 and or FAS. And stain, there is stain called the tunnel stain. It is positive for apoptosis and it is negative for necrosis. And fourth is the DNA electrophoresis, step ladder pattern. That's it for today's class, and we have discussed the big essay topic apoptosis in 2 videos. So make sure you watch all the 2 videos, and thank you. Please subscribe and like my video if you find it useful. Thank you. Thank you so much.